Okay, this video is for Biology 1615 instructors. What we are looking at for the genetics lab is an exercise using a coin flip. And the goal is to get students to understand essentially why did Gregor Mendel use so many pea plants in his experiments. And the idea is that the bigger your sample size is, the closer you will be to your expected frequency. So what students are going to be doing in this lab is each student will have a pair of pennies and they will have a whiteboard, and their goal is to fill out this data collection table. So each student will flip a coin, they'll flip a pair of coins and record their results. So here the result is 1 heads, 1 tails. And they'll use the whiteboard to tally the results, and then they'll use the table to write down the percentages for their results. So I'm going to run through one set so that instructors can see kind of how the flow goes, and then I'll walk you through how to guide students through completing the rest of the table by calculating averages of the 4 sets, averages of the groups, and then averages for the entire class. Then we'll take a look at the data analysis table, which includes comparing the expected and observed frequencies. So each student will flip 2 coins and each event will be recorded. So here I have a result of 2 tails, that's 1 event, and I'm going for a set of 10. That's 1 heads, 1 tails, bam. And if I continued doing this, I would tally the results. Each student would tally their results, and if I just made up some arbitrary values, then I would have something that looked like this for 1 set. They would repeat this 4 times, and then they would tally their results. So in the table is asking students to tally the percentages of their results. So for 2 heads, we got 2 out of 10, that's 20%. 1 head, 1 tail happened 4 times out of 10, that's 40%. And 2 tails happened 4 times out of 10, that's 40%. They would do it for each run, and then they would take the average of the 4 sets, which would be the sum of the values in rows 1 through 4 divided by 4. They would do the same thing here, so that would be 50%. And if they added these all together and divided by 4, that would be 30%. So now we have the averages of the set of 40. Then we want students to calculate the average of their group. So each individual student would share their data for each of these data points. They would add them up and divide by the number of members. So if we had a group of 4 and they each contributed a number here, I'm just gonna make something up. So like 20 plus 30% plus 10% plus whatever. And then they would divide it by 4 if they had 4 group members, or if they had 5 group members, they would divide it by 5. So I'm just gonna put some arbitrary numbers here, 46% and 32%. Then we want students to calculate the average for the entire class. So they have these handy dandy whiteboards where they can share their data with each other. They would provide their sum of values in row 6 for each group. So each group would give these values, and then they would divide by the number of groups in the class. So if we had some arbitrary values here, bam, and 25%. So that's how students would complete this first table. In the second table, students are going to analyze the difference between expected and observed frequencies and write down a positive number. So what they are going to do is they are going to use the data that they got in this table and compare the frequencies, the observed frequencies that they got for each coin flip, the average frequencies of 4 sets, the average of the group, and the average of the entire class, compared to an expected frequency of 25%, 50%, and 25%. So what we want students to do is to give a positive number, which means that they will calculate this by giving the larger number minus the smaller number. So what they're going to do is they're going to subtract the observed or the expected frequency from each other so that they get a positive number. They'll need to use the data that they got on this first table. So the expected hypothesis is this. In the first set of 10, they had 20% heads. So if we subtract 25% minus 20%, we get 5. 1 heads, 1 tail, they observed 40%. That would be a difference of 10. It's an absolute difference. And then in the last one, they got 40%. So 40 minus 25 would be 15. Okay. The total difference there would be the sum of this row. So that would be 5 plus 10 plus 15, which would be 30%. And if I filled that out across for each of the 4 sets, what I would get is, based on the data that we filled out in this table, we would get a difference of 30 minus 25 here would be 5. This would be 10 and 15. So again, they are taking what they have in this set. So set 2, percentage that they observed was 30 for 2 heads. The expected frequency was 25, so the difference is 5, and so on and so forth. We can just fill that out across the table. The goal of this exercise is to get students to realize that as the data set gets bigger, we get closer to our expected hypothesis. And this is exactly why Mendel used so many pea plants to demonstrate the 9:3 to 3:1 ratio of expected frequencies of traits in pea plants.